South America is home to four living camelids, two wild and two domesticated. And for more than a century, scientists agreed on a simple story about how they connected to each other. Both domestic forms, the story went, descended from the same wild ancestor, the Guanaco, while its smaller wild cousin, the Vicuna, had never been domesticated by anyone at all, in any part of the Andes, at any point in history. Scientists eventually compared the DNA of every wild camelid species against the alpaca directly, expecting that long accepted story to hold up under closer scrutiny. Only one wild species turned out to be the real match, and it wasn't the one nearly everyone had assumed for generations of naturalists, herders, and scientists alike. Start with the four animals themselves, because keeping them straight is the whole reason this took so long to sort out properly. The guanaco is the larger of the two wild camelids, standing up to 4 feet at the shoulder and ranging widely across open terrain from Peru down through Patagonia, adaptable enough to survive in everything from coastal desert to high mountain grassland. The vicuna is smaller, more delicate, and confined almost entirely to high-altitude grasslands in the central Andes, prized even in ancient Incan society for a coat of exceptionally fine wool considered more valuable than almost any other natural fiber in the world, historically reserved for royalty alone. The llama, the larger of the two domestic forms, was bred primarily as a pack animal and meat source, capable of carrying substantial loads across difficult mountain terrain where few other domesticated animals could function at all, while the alpaca, smaller and covered in a dense, soft fleece, was bred specifically for its wool rather than any practical labor. Archaeological evidence places the domestication of these animals somewhere between 6,000 and 7,000 years before the present, high in the Peruvian Andes, making South American camelids among the oldest domesticated livestock anywhere in the world, predating many of the domesticated animals more commonly associated with early agriculture elsewhere on the planet. For most of the time since, working out exactly which wild species gave rise to which domestic one seemed like a straightforward question of matching size and fiber type against the two obvious wild candidates. It turned out to be anything but simple once anyone actually tried to prove it. The traditional assumption, repeated across generations of naturalists and herders alike, held that both the llama and the alpaca descended from the guanaco, the larger, more numerous, and more geographically widespread of the two wild species, simply because guanacos seemed like the more obvious candidate for early domestication. The vicuna, by contrast, was long assumed to have never been domesticated at all, its more delicate build and famously skittish, high-strung temperament treated as evidence that early Andean herders simply never managed to tame it the way they had its larger, calmer relative. That assumption held up reasonably well on the surface, reinforced by centuries of herders working directly with these animals and drawing their own conclusions from observed behavior rather than genetic evidence that didn't yet exist. Alpacas and vicunas share a similar, extremely fine wool quality, a resemblance naturalists had certainly noticed over the centuries, but appearance alone was never considered strong enough evidence to overturn what herders and naturalists had already concluded from watching guanacos and llamas behave similarly as social, more easily managed animals suited to close human contact. Untangling the real relationships got dramatically harder after the 16th century, when the Spanish conquest devastated camelid populations across the entire Andean region within a matter of decades. Tax records from the period show herd sizes collapsing by roughly 90% within the first 100 years of Spanish occupation alone, as introduced disease disrupted traditional herding practices built up over thousands of years, and direct competition with newly introduced European livestock like sheep and cattle tore through populations that had been carefully managed and selectively bred for millennia beforehand. In the chaos that followed, llamas, alpacas, guanacos, and vicunas were pushed into far closer contact with each other than traditional, carefully separated herding practices had ever previously allowed, and extensive interbreeding between all four species followed as a direct result, blurring genetic lines that might otherwise have stayed relatively distinct for much longer. By the time modern researchers finally turned to the question of ancestry using actual genetic tools, they weren't just working with four clean, separate populations preserving thousands of years of stable lineage. They were working with centuries of accumulated hybridization sitting directly on top of whatever the original domestication story had actually been, layered so thoroughly that untangling the two would require far more than a single simple genetic test. Archaeologists studying the domestication process itself have found evidence of deliberate camelid management stretching back to the mid-Holocene, with early herders selectively breeding animals for specific traits long before written history existed anywhere in the region to record the process directly. Bone remains recovered from Andean archaeological sites show clear signs of size and structural changes consistent with generations of selective breeding, distinct enough from wild Guanaco and Vicuna skeletons to confirm that domestication had already progressed significantly by the time these populations show up clearly in the archaeological record. Camelids became central to nearly every aspect of Andean life during this period, providing meat, wool for textiles, and reliable transportation across mountain terrain that few other domesticated animals anywhere in the world could have handled, eventually taking on religious and ceremonial significance as well in the societies that depended on them most heavily for daily survival. If you're finding this interesting, be sure to subscribe to understand more DNA from the past. Getting a real answer meant testing the assumption directly rather than relying on body size and fiber quality any longer. The same limited kind of evidence herders and naturalists had been working from for centuries without ever managing to settle the question conclusively on their own. In 2001, a research team led by geneticist Miranda Cadwell published a study in the Proceedings of the Royal Society, examining genetic material from all four South American camelid species together for the first time to finally determine, with actual data rather than inherited assumption passed down across generations, which wild species had really given rise to which domestic one. The team started with mitochondrial DNA, the genetic material passed down exclusively through the maternal line from mother to offspring across every single generation, without exception, comparing sequences across llamas, alpacas, guanacos, and vicunas sampled from populations spread across the region. The initial results seemed to confirm exactly what everyone already believed, and for a moment likely felt like it was simply closing out a settled question rather than opening a new one. 80% of llama and alpaca mitochondrial sequences fell within the Guanaco lineage, appearing to settle the question decisively in favor of the traditional story that had stood unquestioned for well over a century of careful observation. Only a small minority of alpacas carried vicuna mitochondrial DNA at all, and nearly all of those specific individuals were already known or strongly suspected alpaca-vicuna hybrids rather than representative of the species as a whole, which only seemed to reinforce the traditional picture rather than challenge it in any meaningful way. That result should have closed the case for good. It didn't, because mitochondrial DNA only ever tells one narrow part of an animal's genetic story, tracing inheritance along a single maternal line while ignoring the vastly larger pool of genetic material inherited equally from both parents, combined across every generation. Cadwell's team pushed further, examining four microsatellite markers, short, highly variable stretches of nuclear DNA inherited from both sides of an animal's family, rather than just the mother, including two specific markers whose size ranges didn't overlap at all between wild vicuna and wild guanaco populations, making them unusually reliable and precise for telling the two species apart with real confidence rather than probability alone. The picture this second, more complete type of DNA evidence produced looked nothing like the mitochondrial result that had come before it. Alpacas showed strong genetic similarity to vicunas at these markers, and llamas showed strong genetic similarity to guanacos. The exact opposite pairing, the mitochondrial data alone had seemed to support just moments earlier in the same overall study. The The same analysis also picked up clear evidence of bidirectional hybridization running in both directions between the wild and domestic populations, further complicating any assumption that these four species had ever stayed cleanly, permanently separated from one another since domestication first began thousands of years earlier. Subscribe to not miss the next one. Reconciling those two contradictory results required combining both types of genetic evidence together rather than trusting either one on its own in isolation. When Cadwell's team ran a combined analysis using both mitochondrial and microsatellite data together on a subset of samples, the microsatellite interpretation held up as the far more accurate picture of true ancestry, while the mitochondrial signal turned out to be misleading precisely because of the extensive hybridization that the Spanish conquest and centuries of subsequent interbreeding had introduced into alpaca maternal lines specifically. A relatively small number of historical crossbreeding events between alpacas and Guanaco-descended llamas, repeated and compounded across many successive generations, had been enough to gradually replace much of the alpaca population's original maternal genetic signal with Guanaco-derived mitochondrial DNA, even while the much larger, more stable nuclear genome continued to reflect the species' true ancestral connection to the vicuña, sitting quietly underneath that surface-level maternal noise. The alpaca, the study concluded, genuinely descends from the vicuña, not the guanaco, directly reversing more than a century of accepted assumption that had been built entirely on watching which wild species seemed easier to tame, rather than actually reading the genetic record directly. That conclusion carried real taxonomic weight well beyond simply settling an academic debate among specialists. Since the study confirmed the alpaca's true ancestry traced directly to the vicuna rather than the guanaco, researchers reclassified the species accordingly, moving it entirely out of the genus Llama, where it had been grouped alongside the llama and guanaco for generations of scientific literature, and into the genus Vicuna instead, alongside the vicuna itself as its closest living relative. The alpaca's scientific name changed from Llama pacos to Vicuña pacos, a formal, permanent acknowledgement that genetics, not outward appearance or long-standing historical assumption, determines where an animal actually belongs on the family tree once the real evidence finally becomes available to check against. The vicuna and guanaco lineages themselves are estimated to have diverged from a shared camelid ancestor somewhere between 2 and 3 million years ago, long before either wild species had ever encountered a human being, meaning the alpaca's true wild ancestor had been sitting in a completely different genus than everyone assumed for the entire span of recorded scientific classification up to that point, unnoticed simply because nobody had the tools yet to check. The correction mattered for reasons that reached well beyond taxonomy, sitting quietly on paper in a scientific journal. Vicuna fiber remains one of the most valuable natural textiles in the world, prized for its exceptional fineness and warmth, and historically restricted to Incan royalty for exactly that reason. And conservation programs managing wild vicuna populations depend heavily on understanding exactly how much genetic exchange has occurred, and continues to occur even today, between wild vicunas and the much larger population of domestic alpacas raised specifically for their wool across South America, and, increasingly, other parts of the world entirely. Breeders, working to preserve or enhance fiber quality in modern alpaca, herds now have a genuine genetic basis for understanding which bloodlines carry the strongest, most direct connection back to the vicuna ancestry responsible for that prized fiber quality in the first place, rather than relying on visual judgment and inherited tradition alone, the way herders had for thousands of years before any of this genetic evidence existed to guide their decisions. Modern alpaca farming has since spread well beyond South America entirely, with commercial herds now established across North America, Australia, and parts of Europe, all ultimately descended from the same vicuna lineage. The 2001 study finally identified, meaning every alpaca raised anywhere on Earth today, regardless of how far removed geographically from the Andes, still carries that same corrected genetic story inside it. Conservationists, managing wild vicuna populations, some of which had been hunted to the edge of extinction earlier in the 20th century for their valuable fiber, before slowly recovering under protected legal status across several Andean countries, also used this genetic understanding to monitor how much domestic alpaca ancestry has crept back into supposedly wild vicuna herds through the same kind of ongoing, low-level interbreeding the original Cadwell study first documented running across all 4 camelid species simultaneously. Peru, Bolivia, Chile, and Argentina now coordinate vicuna conservation efforts specifically around maintaining genetically distinct wild populations, using DNA testing methods directly descended from the same techniques Cadwell's team first applied in 2001 to screen individual animals before they are included in protected breeding programs, ensuring that conservation resources go toward preserving genuinely wild vicuna lineages rather than animals whose ancestry has already been diluted through generations of contact with domestic alpaca herds grazing nearby. Line up everything the genetic evidence actually revealed, and the story of South America's domestic camelids turns out to be considerably more tangled than the simple single ancestor story repeated for well over a century across generations of naturalists, herders, and textbooks alike, each one confidently passing the same unproven assumption onto the next without ever putting it to a real test. Both the llama and the alpaca were traditionally assumed to trace back to the exact same wild ancestor, the guanaco, with the vicuna dismissed entirely as a species nobody had ever managed to tame across thousands of years of Andean herding history, its wildness treated as simply part of its nature rather than a question worth investigating further. The truth split that assumption directly down the middle, confirming half of it while completely overturning the other. The llama really did descend from the guanaco, exactly as tradition had always held, with the DNA evidence lining up cleanly behind centuries of accumulated observation and confirming that at least one half of the old story had been right all along. The alpaca did not. It descended from the vicuna, the one wild camelid assumed for generations to have no domestic descendants at all, its true ancestry hidden for centuries beneath a layer of maternal genetic noise introduced by repeated hybridization following a population collapse that very nearly erased all 4 species from the historical record entirely within a single century of Spanish conquest. Only one wild species ever truly matched the alpaca's genome once researchers finally looked past the single misleading strand of DNA that had pointed everyone in the wrong direction for roughly 100 years of otherwise careful scientific observation, and it took reading the animal's entire genetic story, both the maternal line and the far larger nuclear genome surrounding it, rather than just the smaller, more easily misread half, to finally prove which ancestor was real and which one simply looked the part from the outside the entire time.